Hi everyone, welcome back to Dr. Han's Classroom Pro. Now let's talk about the basics of viral infections today. This is a quick review of basic viral structure and its cycle, so let's get started. Now viruses are not technically considered to be living organisms. They are obligate intracellular parasites that require a host to propagate. However, they contain either DNA or RNA genetic material and are surrounded by various types of protein. Now, without a host, viruses are not able to carry on metabolic processes as they lack all the essential cellular structures. In other words, viruses are nothing without us. Now, from a medically related view, it is natural to think of us, humans, as the virus host. Now, but in fact, viruses can infect all known forms of life, including plants, animals, and even bacteria. Now, the best treatment for viruses is the host immune system generating proper humoral and cellular responses to fight them. Now, if you don't remember immunology, I have a whole course uploaded on my channel and you can learn more about that topic. As a side note, the famous CRISPR-Cas genetic engineering tool was originally discovered as the immune system in bacteria, which they use to fight bacteriophage. Check out my CRISPR video if you want to learn more about that. Now, viruses do come in many different shapes and sizes. Plant viruses can be in a tubular shape. The bacteriophage looks like a lunar lander. Mammalian viruses are usually more spherical. Here we are focusing on the mammalian virus's basic structure. Viruses could have RNA or DNA as genetic material, and it is packaged within a protein capsid. The term nucleocapsid describes the viral nucleic acid plus the surrounding capsid. Some viruses have an envelope, which is a lipid-containing membrane and is acquired during the budding step from the host cell during the viral cycle. The envelope is also covered with proteins that can interact with host cell membrane receptors for binding. Now, so we can call all the viruses without the envelope the naked virus. Now, here is the visual comparison between the naked virus and the enveloped virus. Now, remember, naked virus is basically the nucleocapsid in enveloped virus. We can classify major groups of viruses as either DNA or RNA viruses. RNA and DNA viruses can be single-stranded as ssRNA, ssDNA, or double-stranded dsRNA or dsDNA. They can carry either the positive sense or the negative sense of the nucleic acid. Shape-wise, the genomic material can either be linear or circular. David Baltimore classified viruses into 7 groups. The groups are based on genome type, mode of replication, and transcription. But regardless of which group viruses are in, they all have the basic steps in their viral cycles. It begins with binding to the receptors on target cells, entering into the cell, uncoating, replications of their genetic material, assembly of the viral proteins and genetic material, and release of the newly formed progeny of viruses to infect another cell. Now, of course, during this process, the host immune system should kick in to stop this perpetual cycle. Now, let's look at some of these steps in greater detail. Now, during the attachment step, the viral protein looks for a specific receptor on the plasma membrane to attach itself to. For example, influenza's HA protein binds to sialic acid on cell surface, and HIV GP120 attached to CD4 receptors on T cells, and SARS-CoV-2 spike protein binds to ACE2 receptors on epithelial cells. After attachment, the entry step can vary slightly among different viruses. naked virus directly penetrates or enter through endocytosis. Enveloped viruses enter through membrane fusion and as well as endocytosis. After the entry, capsid shall disintegrate and release the viral contents into the cell. The replication step is where the major differences are among all the 7 groups of viruses. Now for DS or double-stranded DNA viruses, they are more straightforward. Double-stranded DNA is directly replicated into a new copy of double-stranded DNA. Single-stranded DNA viruses need to generate a new single strand of positive sense DNA from the double-stranded DNA. Double-stranded RNA viruses generate a new copy of double-stranded RNA from their messenger RNA. Positive strand single-stranded RNA viruses first use RNA-dependent RNA polymerase to generate the negative strands of the RNA, then regenerate the positive strand for the new viral particle. Negative sense single-stranded RNA viruses such as influenza virus first used RNA-dependent RNA polymerase to transcribe its negative sense RNA to positive sense and transcribe one more time back to the negative sense. The process for single-stranded RNA retrovirus, such as HIV, is more convoluted. The RNA is first reverse transcribed back into single-stranded DNA, then DNA polymerase kicks in to make a double-stranded DNA, and lastly, transcribed into positive RNA. Now lastly, we have DNA viruses that contain gaps in complete DNA sequences. Now these DNA first utilized host DNA repair mechanism to generate a full double-stranded DNA and goes through steps similar to retrovirus by first generating positive single-stranded RNA, reverse transcribing to single-stranded DNA, then duplicating back to gapped DNA. The hepatitis B virus is an example of gapped DNA virus. The viral particle assembly locations vary depending on how the virus replicates its genetic material. DNA viruses are assembled in the nucleus and released into the cytosol. RNA viruses develop in the cytosol. Generally speaking, animal viruses need more time to develop and mature in the cell. After viral particle assembly, enveloped viruses are released by budding and take a part of the cell membrane along with them to form the viral envelope. Naked viruses are extruded from cells by exocytosis. They can also release themselves by lysis of the host cell. Now this completes the viral cycle. Some viruses, however, can enter a latency stage during their viral cycles and remain dormant in cells. For example, chickenpox and herpes viruses lay dormant by hiding in host cells. Now in this case, they do not become incorporated into host genome. However, some viruses, such as HIV, become integrated into the host chromosome as a provirus. This integration is permanent, and when this cell undergoes mitosis, the descendants of the cell will all carry the provirus gene. Now that is all on the brief review of the basis of viruses. Leave me a comment if you have any questions, and I will see you in the next video. Take good care. Bye.